After the data, what we have to do with the data which we have collected and how to collect data, what are the methods. Let me share the PowerPoint first. So it will be about the biological variables, rigor, reasoning, and resource authentication. So first thing we need to understand in this lecture about the how we have to select the biological variables, what is rigor of the research, what is the reasoning in the research, and how we can authenticate the resources which we are using in the project which we are working on. So first thing is biological research variables. So variables are any characteristic, either there may be a number, quality, quantity, name, age, BMI, any type of data which we are working on to compare between the control and the disease or any two groups or two criteria. They are called as biological variables. So they may be either measurement variables, which are numerical values like height, weight, number, blood pressure, BMI, or nominal variables like categories or names. Then ranked variables, it may be the position or order of the patient or any of the variable which we are using. Then independent variables are those which we use to do something like monitor or manipulate or change. Like, for example, if we are working with the cells and we are treating the cells with some cytokines or anything which increase inflammation like LPS or anything. And if the expression of any gene is changing, since we are changing it, so this is called as independent variable. It is not dependent on cell line itself. We are intervening to change it. Dependent variables are those which are getting changed due to our intervention and measured after our intervention. Confounding variables, mostly they are in clinical research and sometimes in basic research also. Like in, for example, in clinical research, if you are working with the BMI, body mass index, that why the patient are getting obese, what are the factors, and you are looking for the alcoholism, smoking, diet chart, all those things. Even you are looking for the exercise, but you are not looking for their sleeping habits, how they are traveling, or are they having a active life or sedentary life. So those variables are called as confounding variables, mainly which we ignore or they are hidden and we are not knowing about those variables and they may be hidden. Other example, like if you are looking for the hypertension and you are looking for salt intake, water intake, body mass index, weight, and all those things. But you miss the thing that they are taking some medications for some other disease or they are taking the over-the-counter medications. So those are called as confounding variables, means the variables which affect the disease process and the data collection, and we don't know about that. Means unknowingly we are ignoring it, but they may affect the data quality. They are called as confounding variables. So example of biological variables are like age, sex, body mass index, blood pressure, disease severity, and why the biological variables are important to consider. Why we should consider multiple variables in a research. So first thing is to understand the disease. Like any disease which is we are working on, whether it is more prevalent in male or more prevalent in female, or Americans, African Americans, Asians, or any other Caucasians, which race it is more prevalent, or it is more prevalent in aged individual or middle age or younger individuals or children. So it is very important to understand the biological variables regarding the any disease process. Then developing treatment. So if we know that this certain disease is associated with a certain variable and it is more prevalent in some population, or it is dependent on the gender of the individual, then we can develop the treatment accordingly. That is called as personalized medicine. So for personalized medicine, it is important to understand that how the disease process is going on, whether it is variable dependent or variable independent, or it is generalized. Then improving public health. So improving public health means we can have a personalized medication or generalized medication. So if variable which is associated with the disease is not associated with either sex or gender or age or feeding habit or any other variable, then we can have a generalized treatment. Like for example, COVID-19. So recently we have faced it. So COVID-19 was spreading in everyone. Obviously, the elderly and disease individuals were more prone, but we gave the vaccine to each and every person. So that is called as generalization of the inference or whatever we have seen. And based on that, we reach to the conclusion that we have to immunize everyone. That means we can improve the public health in general. So for that, these variables are very important. There are some NIH guidelines that what variables we should choose. So when we are collecting the data, either in the basic research or in the clinical research, it is important to choose the variables which are relevant to the research. Not like we are collecting all the data, as I discussed in the last lecture. So we should choose the variables on which we are collecting data. It should be relevant to the research question. It's not like we are collecting it, let's do everything, which is not allowed. Then ethical consideration means whenever we are collecting any data regarding any variables, we should always consider the privacy of the individual, that we are not going to disclose it. And whatever we are collecting, the data is as per the rules and regulation and legal things as per the institution which we are working in. So we should always consider the ethical consideration when collecting the data on any variable. Then statistical analysis means whatever the data we have collected, we should analyze that data with appropriate statistical method. So for that, we have to see whether the data is following the standard curve or not. It is normalized or not normalized, whether it is lying within the two standard deviation or not. So there are so many things, means there are multiple parameters before choosing any statistical method. Second thing is whether we are comparing the data between two variables and more than two variables. Another important thing, whether the variables are dependent variables or independent variables. So based on multiple things, we need to choose the method of the statistical analysis. Like if there are two independent variables and we are comparing only in between two, then we can use the t-test. If they are independent plus dependent and we are comparing more than two groups, then we can do the one-way ANOVA. If it is more than three groups, then we can do the two-way ANOVA. Similarly, three-way ANOVA and four-way ANOVA, like that. Then if we are doing population statistics, then we have to use the chi-square method. So, and if we are correlating anything, like we are correlating the weight with hypertension or weight with blood sugar or smoking pattern with blood sugar or hypertension, then we have to do the correlation analysis. So in correlation, which correlation we have to do and what correction we need. So all those things we should consider before analyzing the data. Then coming to the scientific rigor. So scientific rigor means whatever we are doing, either in basic research or clinical research, the method should be applied very effectively. Mostly it is related to basic science research, like if we are working on any project and we are treating the cells or we are working on the tissues collected or we are working on the animals. So what experimental methods we are using, whether those experimental methods justify and explain the cause or the reason which we are looking for. It's not like that we know so many methods like immunohistochemistry, histology, staining, basic staining, then some advanced staining, multiomics, RT-PCR, ELISA, Western blot, and we are doing all of that for everything. So it's not justified. We have to look for whatever our hypothesis is, and based on that hypothesis, what experimental methods are needed and whether those experimental methods are in depth or not. It's not like we are just doing superficial methods. If we want to look for the gene expression in a population and we are just doing RT-PCR, which is not justified. So for that, we have to look for the sequencing. So what are the basic genes change? So RT-PCR gives us the gene expression change, but not for everything. So for in population, we should go beyond that. So whatever the method we are using, it should be in depth, and that thing is called as rigor. So rigor depends on multiple things. That what is the logic of using any method? So why, whatever the method we are using, what is the logic behind it? Why we are using it? What is the rationale for that? Then error analysis. So when we are collecting the data, always there will be some bias and some errors. So we should always be concerned for errors and bias in the research. So if we are collecting any data, we should know that what errors may happen. Like as a PI, if somebody is collecting the data and they are telling their postdocs or medical students or residents that collect this such and such data from all the patients. And every day, the medical students or residents are changing their duties, and so many persons are collecting it. So in that, there may be a observation bias or collection bias. Means some person may collect in a different way, some person may collect in a different way. And there will be a, like if you are saying the pain severity, we are collecting the data on the pain severity. I can say that this pain is 5 out of 10. And you may say that maybe this is 4 or 6. So this is called a error in the collecting data or bias. So we should know that these things may happen and what are the measures to mitigate these things. How we can modify this, how we can correct it. Then third thing is intellectual honesty, that whatever we are collecting should be as it is. We should not give input from our side. So whatever it is, it is. We should not manipulate it. We should not give our input from our side. Whatever the patient is telling or whatever the results we are getting from the wet lab experiment, it should be that. We should not manipulate anything. Then probability. Probability is the thing that if you are hypothesizing something in the clinical research or basic research, what is the probability of that thing being to be positive? If you are hypothesizing something which is not possible, so in research we say that you are cooking the pie in the sky, means this is not possible. How you can start the research on this hypothesis? So before starting the research, you should look for the literature, not only in the subject or stream which you are working on, with the other streams also. Whether is there any literature regarding that gene or protein or anything. And if there is something, then there is a probability that the same phenomenon, same changes may happen in your system also. And if there is no literature and if there is no supporting data, and you are saying, no, it will happen. So the probability of happening that will be very less. And that may lead to the negative output of the experiment, which is not correct. So we should look for the probability, that how much probability is that to be this hypothesis as positive or whatever we are thinking is correct. Then statistics, as I discussed, we should already have a plan that how we will be analyzing the data, what statistical method will be used, who will work on the statistics, whether the person who will be working has the knowledge of it or not, he is certified or not, all those things. Then experimental redundancy. Experimental redundancy means we should not do the experiment which has already been done. Like if you are exploring something on a particular gene called X, and you find a paper where they have done RT-PCR and immunostaining, and you are going ahead with the Western blot, ELISA, or metabolomics, proteomics, or something, and you are also doing the RT-PCR and immunostaining, which already has been reported. This is not allowed. Whatever has been done, you have to take the reference from that this thing has been done. Based on that, we've developed our hypothesis. But we cannot do the same thing again and again. So we should avoid the experimental redundancy. So rigor, as I discussed, methodological integrity, whatever the methods we are using in the research, we should know that these are relevant to the research question. Ethical consideration, data collection and analysis, Reporting and transparency. So reporting and transparency means whatever the data we are collecting, analyzing, should be reported in a transparent manner, so that if somebody is having that data, can replicate the same results or same trend whatever you are having. So whatever the data we are collecting should be transparent, and it should be reported in a transparent way without any modification or changes. You may mine the data, but you cannot change the data. Rather in different research types, so quantitative research and qualitative research. So quantitative research means it uses numbers and statistics, like whatever we are doing in the lab. We are testing one hypothesis. Are making a hypothesis, then we are doing multiple experiments, and we are testing the hypothesis. However, qualitative research means when we are not using the numbers, we are looking for some written things, oral videos, or audios, and then we are making some inference out of that. Like if we are doing some poll analysis and multiple persons are giving their consent or their views, and then we are extracting a hypothesis out of that. Okay, so these persons are having this view, and out of 100, 90 are having this view, so this is correct. So we are making our conclusion based on the views of other person. We are not collecting any data in that. So that is called as qualitative research. So in quantitative research, rigor is often associated with concept like validity, generalizability, and reliability. Means whatever the data we are collecting is valid, is reliable, and it can be generalized to the population. It should not be for a selective group of population, like only for males or only for females, only for kids or only for elderly or certain race. It should not be. It should be a generalized output. Same thing in qualitative research. There are three components: trustworthiness, credibility, and dependability. So whatever you are, either you are taking audio interviews or video interviews or written contents, you should have a trust in between you and the person from whom you are taking it. Then credibility and dependability. So whatever you are reporting, it should have a credibility, and you should be responsible for that, whatever you are reporting. And whatever the output is should depend on the data which you have collected. So that's called rigor. Benefits. What are the benefits of rigor? Trustworthiness. It will be improve the validity, enhance generalizability of the data and output, and then reproducibility. Means whatever the data you are reporting, either from clinical research or basic research or the population research, it should be reproducible. Means if someone else is working on it, they should get the similar results. Then this one is reasoning. So reasoning is the logical process of drawing conclusion, making prediction, or developing explanations. Means whatever we are doing in the research or whatever the hypothesis we are working on, we should have a rationale for that. That whatever the things we will be doing or specific aims we are working on to test our hypothesis should have a rationale for that. And what is the reasoning behind that? And that reasoning and rationale should depend on the existing literature. May not be in your field, maybe in a different system of biology or a different field, but it should have some rationale behind it. You should not just randomly pick a gene or protein, okay, I want to work on that in this disease, but that gene or disease are not related at all in any way. That means this is wrong. We should have some rationale behind that. It helps in researchers to interpret the data because you have a trend like gene X is related with some disease in cardiovascular system, for example, and you are working in musculoskeletal, and obviously the body is the whole unit, so the systems are related to each other. If this gene is involved in cardiovascular, it may involve in musculoskeletal, and you are getting some results out of it. So the previous results which you have read in the literature will help you to analyze your results also, whether you are getting a negative result or you are getting a positive result. Means same as the previous which has been reported, or the opposite of what previously has been reported. So it depends. You may get a positive or negative, but it should be related to that. It's not like something you are getting which is not related to anything. It will refine your understanding and communication of the data which you have collected to the scientific community or as well as general person population. Then reasoning involve both deductive and inductive reasoning. So what is deductive and inductive reasoning is? So inductive reasoning is the space start with a specific observation and moves to a general conclusion. Like we are reading the literature, we are making some hypothesis, and we are having some observations from that. Like if you say that all the birds can fly, and if you are seeing a something fly, you will simply say, oh, this is a bird. Because it's a dictum that okay, all the birds can fly. And if you so you have a observation that something is fly, you can say it may be a bird. Or if you are allergic to the food, you can say that if person is eating that food and getting allergy, that you can say, okay, this person is allergic. Because we know that that food has some allergens. So we know that. Deductive is have a general premise and moves to a specific conclusion. You are saying that all the swan are white, and when you observe a swan and say this is white, because we have a general thing that all the swans are white. So there is a very thin line, but there is a difference. In the inductive reasoning, we generate the hypothesis and then prove it. In deductive reasoning, we observe it and we generalize it. Then abductive reasoning is identifying the most likely explanation for this set of observation. So if you are looking something in multiple places or multiple observations, and then you are setting a hypothesis for that. So what may be the explanation for the same kind of result in so many persons? And you, based on your thinking, on the literature, you are making a observation or explanation. That is called as abductive reasoning. Then analogical reasoning involves the drawing the conclusion by comparing similar case or situations. So there are similar cases at multiple time points, and then you are drawing a conclusion from that. So that is called as analogical reasoning. Then coming to the importance of reasoning, it will help us interpreting the data. It will refine our understanding of the research or any disease process or any behavior going on in the general public or general population. Then communicating the findings. We can easily and effectively communicate the findings, whatever we are getting, with the help of reasoning and building credibility. So if whatever you are reporting is credible based on the evidence and data, that will enhance your credibility, that whatever your university or you are reporting is correct, and people will follow that. Second, like in basic research, if you are reporting that this is a target for this disease, and if we are targeting this specific gene or protein with a drug, its expression is going up or down based on your research. And this has these beneficial effects. And if I am doing that and I am not getting that answer, or 10 more persons are doing the same thing and they are not getting the results what you are reporting, that means either me or you, both are losing the credibility. Means the credibility of research is gone. So whatever we are reporting, the other person should be able to replicate it. They should get the nearly similar findings. It may not be exact same, like if you are getting a 7-fold change using a cell line in any gene expression. Maybe if I am doing the same experiment, using the same cell line, treating the cell line with the same reagent which you have treated or I have treated, and I am getting a 6.5-fold or 7.2-fold. So it is increased, but it is not necessary that I will get the same 7-fold, because there may be multiple confounding factors. The cell line which you are using have a different progeny, which I am using have been cultured multiple times, so I have a different passage number. The cell line have changed the phenotype, their characteristics. So those things may change the result, but it should not change the trend. Like if expression is increasing, everybody should get it increasing. Not like that I am getting increase and you are getting decrease. So trend should be same, may not be the exact same number. So that is called as credibility of the research. Then coming to the examples, it helps in developing a hypothesis, it helps in developing it, testing a theory, and then it helps in interpreting the quantitative or qualitative data which we are collecting. Sometimes we can use both the reasoning, inductive and deductive also. So initially we can use the inductive reasoning for making a hypothesis, and then we are testing the hypothesis. That is deductive reasoning. So it may go side by side or before and after and in parallel also. Then coming to the resource authentication. Resource authentication means whatever the resources we are using in our research, whether we are using cell line, antibody, primers, proteins, or anything, we should know from where it is coming, who is providing it, whether they are getting the same antibody if we are ordering it again from the same clone of cells or same clone of animals. The clonicity is same or different. And if it is different, we should note it down. So that is called as authentication of the resources, whatever we are using. If we are using any instrument, from where that instrument is coming, whether the settings on that instrument is correct or not, whether the rule, the specifications, the scale of the instrument is correct or not. If we are using a refrigerator or a thermocycler or a centrifuge, which is at 4 degree, whether that apparatus or equipment is maintaining the 4 degree or not, or if it is -20, whether it is maintaining -20 or not, how we know it. So we should, obviously they say that it is maintaining -20, but how we know that? So we should put a external thermometer. And if our external thermometer is giving the same temperature which is specified in on that equipment, then we will say, yes, this is authenticated. Same thing for centrifuge. If you are saying that I am centrifuging this sample at 4000 RPM, how you know that the centrifuge is running at 4000? Obviously, the panel says 4000, but how you know that? That panel is giving us the correct value. Maybe panel is malfunctioning and rotor is spinning at 6000 and panel is giving us at 4000. Or panel is giving 4000 and rotor is only 2000. So there are methods where we can authenticate these readings, and each and every lab should follow that. Same thing antibody. If they are saying, we are seeing on the vial of the antibody that it is raised in rabbit and it is against human. How you know that? So you look for the SGS sheet, data specification sheet, that what they have written in that is correct. And the same thing is on the vial of the antibody. So if they are matching and if there is any mismatch, you have to call to the company from where you are ordering it and ask them that why there is a difference in that. So sometimes we have to believe on that whatever is written on the SDS sheet and the vial of the antibody is same, means it is the same, correct thing. We cannot test for each and everything. But if needed, and if there is this disparity and you think that there may be something wrong or antibody has got precipitated, color has changed, then you have to check it first. You have to check that antibody in your lab using the Western blot or electrophoresis gel, whether the antibody is binding to the same target which it is intended for. Or you can do the immunohistochemistry or immunostaining to check for the antibody whether it is working in the same way which it should be. If it is not, mean there is something wrong. Same way for the primers. You may design primers or you may order the pre-designed primers. So if you are ordering the primers or pre-designed primers, you should check them whether they are correct or not. So that is called as resources authentication. So it will help to ensure the integrity and validity of the materials, resources, and the data which we are using in the research. It improves the reproducibility and reliability. That means whenever we are publishing a manuscript out of the data which we are getting, so we will be including the catalog number of the antibody or any resource which we have used, maybe a chemical or a buffer or anything. So we have to include the catalog number and the vendor name, that we got this antibody or any chemical or any buffer or any reagent from this catalog number was there, and this is the vendor. So if I want to do the same thing to check whether you have done it correctly or not, I need to order the same antibody from the same vendor using the same cell line or same tissue. And then if I am getting the same results, means whatever you have done is correct. So that is called as reproducibility. And that depends if you are including all the identification of each and every reagent in your manuscript, so that I can order the same thing. So that will increase the reliability also. Whatever you are reporting or I am reporting, they are reliable results. Whether it is clinical research or basic, doesn't matter. It's about verifying that resources is what it is claimed to be, as I discussed earlier. It is crucial for scientific rigor and transparency. So if you are using multiple methods like Western blot for protein expression, then you are doing immunostaining for the protein expression, and whether this antibody is binding to certain protein at a certain location, you are doing immunoprecipitation or chip assay. And if that antibody is behaving in the same manner in all four methods, so that means you are using a rigor, you are justifying the use of that antibody and getting the same result in four different techniques. That means your results are transparent. There is no manipulation, no alteration, nothing from your side. It also helps in preventing the errors. So if you are using an antibody or primer or any chemical or any buffer, and you are not looking at the STS sheet and whether it is matching with the label of the bottle or vial, there may be a chance that by mistake you have picked up a wrong vial, or they have labeled it wrong. Like in the primer, they will paste the label on the vial, and maybe they will have pasted the wrong label on the vial. And if that primer you are using for your experiment, you will totally get the wrong results, or totally results which are not in concordance with anything. So you have to check it. Whatever the sheet they are giving, it is matching to the label of the antibody or primer or any reagent, chemical buffer, anything. So it helps in preventing errors if you are authenticating it, if you are checking it thoroughly. It also helps in FAIR principle. So FAIR principle stands for findable, accessible, interoperable, and reusable. Means you can find the antibody. If you are using, I can find it. It is accessible. It may be possible that the vendor from where you are ordering it had discontinued it. So they will say that we have discontinued this antibody production. But still I can get that information that whatever you have used 3 years back or somebody has used after 10 years before has been stopped. They are not producing it anymore, or they are not supplying it anymore, but it was there. So the researcher who have used it is correct in their manuscript. So it is accessible. Then interoperable means we know that what is the interpretation of that. So we should know that. And then it should be reviewed. Then I discussed antibody for all of them. So with this, thank you for your time, and any question? Any question? Any doubt or anything which I